Question number 1. An enzyme which is not used in rDNA technology is: first, reverse transcriptase; B, DNA polymerase; C, restriction endonuclease; D, DNA RNA ligase. So reverse transcriptase is required for conversion of RNA into chimeric DNA. DNA polymerase and C, restriction endonucleases is been required for fragmentation of DNA. And option D, which is RNA ligase, is for connecting the RNA fragments or joining the RNA fragments. So option B, which is not used, has no application in rDNA technology. So option B is the correct answer. It is not used. A DNA polymer is not used in rDNA technology. Rest all three are used in rDNA technology for some or the other purposes. Question number 2. For recombinant DNA technology, the host should be A, pathogenic; B, harmful microorganism; C, harmless microorganism; and D, difficult to cultivate. If the microorganism is pathogenic or harmful, it will lead to disease or any pathological changes. Any pathological changes can be there. So to avoid this, pathogenic harmful microorganisms are not used in rDNA technology as a host. Similarly, if they are difficult to cultivate, then the amount of expression will be less. The amount of expression will be less if it is difficult to cultivate. So option D is wrong. Option A and B are also wrong. So option C, which there should be harmless microorganism for the rDNA technology, for the host should be an harmless microorganism if it is to be used as a host in rDNA technology. So option C is the correct answer. Question number 3. Restriction enzyme belongs to class what? Restriction enzyme is being used in rDNA technology for fragmentation of DNA. For fragmentation of DNA we use restriction enzyme. Since it cleaves the DNA, it is also known as nuclease. So restriction enzyme belongs to the class nuclease. So option D is the correct answer for question number 3. Question number 4. The transfer of genetic material from one bacterium to another without vector is termed as. So first let us rule out these two options: translation, transcription. They are being used in protein synthesis, and this occurs in vivo. So these two are wrong. Transduction is with respect to bacteriophage. So transduction requires a vector. So the only option that is transformation requires no medium for transfer of genetic material from one bacterium to another, so without a vector. So it is termed as transformation. So option A is the correct answer. Question number 5. A recombinant DNA molecule can be produced in the absence of the following: A, E. coli; B, DNA ligase; C, DNA fragments; and D, restriction endonuclease. Restriction endonuclease acts on DNA to form DNA fragments, and these DNA fragments are being then acted upon by DNA ligase to form a DNA molecule. So a recombinant DNA molecule is obtained after this process with respect to all this. So for the formation of rDNA molecule we require these three enzymes. So the odd one out is E. coli. So E. coli is the correct answer. Question number 6. Of the following, which phenomena is involved in the synthesis of cDNA? cDNA is also known as complementary DNA. So this complementary DNA is been obtained by process of reverse transcription, which we call it as teminism. So teminism is the correct answer for this. Teminism Termination wrong. Transformation is conversion of one bacteria into the another in terms of characteristics. Transduction with respect to bacteriophages. So this two options are also wrong. So terminism is correct. So terminism is the process of obtaining DNA, complementary DNA from RNA by reverse transcription. So option B is the correct answer. Question number 7. The enzyme used in PCR. PCR is polymerase chain reaction. So polymerase chain reaction. So this polymerase chain reaction technology uses enzyme DNA polymerase. Reverse transcriptase, RNA polymerase, prokinase are not used in PCR. So we use DNA polymerase. Option B is the correct answer. Question number 8. The incorporation of rDNA into bacterial cell is generally done by A. Centrifugation, B. PCR, C. Autoradiograph, and D is electroporation. So centrifugation will separate, PCR will amplify, autoradiograph will give you imaging. So electroporation, where pores are being created with respect to electric currents. So from this pore, this is rDNA, incorporation of rDNA occurs. So option D is the correct answer. Question number 9. Cloning is possible. A. In vivo means inside, in vitro means outside, both in vivo and vitro, and D is in microbes only. So it is possible outside as well as inside, since cloning can occur naturally as well as artificially. So it is possible in vivo and in vitro as well. So option C is the correct answer, which states that both in vivo and vitro. Question number 10. PCR technique is best for option A. Protein synthesis, option B. Amino acid synthesis, C. Lipid synthesis, D. DNA amplification. Protein synthesis is natural process, amino acids as well, and lipid synthesis, all three are natural process. This is an artificial process where the DNA fragments are being amplified for a specific segment. For a specific segment they are being amplified. So this amplification is nothing but concentration, is concentration of the DNA fragment. So option D is the correct answer, DNA amplification. Question number 11. In prokaryote, jumping genes can jump from. So jumping genes are also known as transposons. Option A. Plasmid to chromosomal DNA, plasmid to ribosomes, plasmid to cell membrane, plasmid to non-genetic double-stranded RNA. So first of all, plasmids, these are genes which can be transferred from one genetic material to another genetic material only. So option A, which states that plasmid to chromosomal DNA, is correct. Ribosome is not genetic, cell membrane is non-genetic, and this also is non-genetic. So this all are wrong. This option A is right because it's from one genetic material to another genetic material. So option A is the correct answer. Question number 12. Which one of the following is the significance of transposon? Transposons are nothing but jumping genes. Since they are genes, they are just specific segments of DNA. So first option helps to remove a genome. Not possible because it is an entire DNA. It is just a segment. So first option is wrong. It helps in purity of organism. No, wrong. C. It helps to restructure a genome. And D. It helps to transfer an entire genome. Entire genome cannot be transferred since it is only a part of a DNA. So it helps in restructuring of genome. So it helps in restructuring of a genome. So option C is the correct answer. Restructuring means transfer of gene segments. Transfer of gene segments is restructuring. So option C is the correct answer. Question number 13. Plasmids can occur in. Plasmids are nothing but extra chromosomal DNA. So this extra chromosomal DNA is circular in nature. So it is found in prokaryotic organisms, and this can be a bacteria or a fungi. So option D, which is a yeast, which is a fungi, and bacteria is the correct answer. So that is D is the correct answer. Question number 14. An antibiotic resistant gene in a vector usually helps in the selection of recombinant cell. Recombinant cell selection does not depend upon the antibiotic resistant gene. Competent cell also, and transformed cell is the cell which has been transformed into with new characteristics. So first three options are ruled out. Chimeric DNA, also known as cDNA, which has been obtained by a process known as teminism, that is reverse transcription. For this process we require for antibiotic resistant vector usually helps in the selection of this DNA. So what kind of DNA we prefer in a vector? So antibiotic resistant gene. So option D is the correct answer. That is chimeric DNA. Question number 15. Cosmid contains. First of all, cosmid is a constructed vector. It is a constructed vector which consists of lambda phage DNA with plasmid. So cosmid is plasmid plus lambda phage DNA. So it will contain fragments of lambda phage DNA. It will not have fragments of plant virus, animal virus, or plant and animal virus. So option A is the correct answer. Question number 16. A phage is virus which eats upon. Phage is the full form is bacteriophage. So bacterio means bacteria and phage means to eat upon. So it eats upon bacteria. So option C is the correct answer for question number 16. Question number 17. M13 bacteriophage infects. M13 is not a lambda phage. It is not a lambda phage. It is different from it. It has 72 nucleotides. And it infects mainly bacillus species. Bacillus means rod shaped. So which can be E. coli, salmonella and all, as well as agrobacterium species. So all these options are included in bacillus species. All these options are included in the bacillus species. So this all three are rod shaped bacteria. So there is bacillus. Option D is the correct answer, which states that bacillus species. So option D. Question number 18. DNA of M13 bacteriophage is. M13 bacteriophage is different from lambda phage in terms of DNA. So it has a circular DNA which is single stranded DNA. So circular and single stranded DNA, and with 72 nucleotides. So option A is the correct answer for question number 18. Circular and single stranded. So option A is the correct answer. Question number 19. Daughter bacteriophages are released from the bacterial cell by the action of. So this is a bacteriophage which, when infect a bacteria, incorporates its genetic material into the bacteria. Then the small viroids develop inside the bacteria, which release an enzyme which acts on the bacterial cell and causes the release of bacteriophages again. So it is endonuclease and exonucleases acts on the nucleic acid. So this both options are wrong. Isomerase is responsible for isomerization, so it's also wrong. So option A is the correct answer, which is the endolysine. So option A is the correct answer. Question number 20. Restriction enzymes. Remove enzymes remove introns, transcript mRNA, cleave apart foreign DNA and not vector DNA. Correct answer is option D, which states that it cleaves upon foreign DNA molecule. So restriction endonucleases acts on the foreign DNA molecule and not on vector DNA, so that the foreign DNA has a, so restriction endonuclease acts on this, so this part is left on which incorporates the vector DNA. So the new characteristics are been incorporated. So option D is the correct answer for question number 20, which states that it cleaves upon foreign DNA and not vector DNA, so that vector DNA gets incorporated into the foreign DNA to bring out the required characters. So option D is the correct answer.